Hello, the Biochemical Society and Portland Press are pleased to welcome you to this webinar, part of our Biochemistry Focus webinar series. Topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support early career development. Each webinar gives you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. Hi everyone, my name is Richard Reece. I'm the Deputy Vice-Chancellor at the University of Kent and a trustee of the Biochemical Society and chair of the Portland Press Board. It's my pleasure and privilege to welcome you today and to introduce today's speakers for what I think is going to be an absolutely superb session. Today's webinar is called How to Deliver Biomolecular Science Honours Research Projects with Limited Lab Bench Time. Lab time and bench science were likely to be limited for many final year undergraduates due to social distancing, potentials for continuing shielding and travel restrictions. Consequently, research projects need to be adapted to this new landscape while still achieving their aims and learning outcomes. This is the second instalment of a two-part series that aims to share the advice and expertise of supervisors and students with experience of such projects, that we hope you'll find the information that we share is useful and perhaps be able to adapt it for your own use. Today we welcome Aysha and Vaz from the University of Leeds, where Aysha is Associate Professor and the Faculty Director of Student Education in the School of Molecular and Cellular Biology, and Vaz is a reader in Human Disease Biology. They'll be talking about the computational projects that they run with a focus on the interrogation of gene expression data sets of cancer patients, statistical analysis, and using R and building up models of cancer disease progression and patient survival. Please make sure that you ask questions during the talk. You can do this and we'll take them at the end of the webinar, but you can send in your questions at any time during the talk. So if you do have questions, please type it into the question box as shown in the image on the screen. I'd now like to take this opportunity to hand over to Aysha and Vaz, who will take us through their presentation.
So thanks everyone for joining, and thank you Richard for the introduction. Both Vaz and I are the computational project that we ran actually with master students over the summer period. But you'll see as we progress through the talk that it will be possible to adapt this shorter. So just to start by providing a little bit of context, we had in April in the Faculty of Biological Sciences approximately 40 master's project students due to start in the research labs conducting a wet lab project. In mid-March, April, to the pandemic, universities shutting down and the government's going into lockdown, we had to position all wet computational data analysis type projects very, very quickly. And our aim really was that we provide students with the opportunity to conduct remote projects so that they could still complete their MSc degree within the usual time frame. The aim was to adapt or replace the existing project, align with an online format, but stay with the same supervisor as far as possible. And the project continued to be...
It's breaking up a little bit, unfortunately. Vaz, might you be able to try taking us through this bit? I realise that's somewhat out of order, but it might help with the sound quality.
Yeah. Okay, so what we had was actually 40 master's project students that were supposed to do research projects in 2020 in our laboratories, and these were for largely all wet lab based projects. So because of what happened with the shutdown of laboratories by early April, this became impossible to deliver. So what we tried to do was to adapt these projects to an in silico format where possible. So to try and keep the same supervisors and adapt the research project to deliver them in silico. So one of the key criteria is to try and ensure that the learning outcomes that link to the degree program are met. and this is training in experimental design, data generation, analysis, and interpretation. And the idea is that this then delivers the value to the student and helps them move forward in terms of their research, their career, and their job prospects in the long term. Yep. Okay. So the identification of key issues in the project was, so the central problem was actually developing and running an in silico research project. And so to state that both myself and Aysha as project supervisors had limited or no experience of such work. Okay. And then, so one of the things actually was to design the project based on prior work in that area. So to make sure that there is a very solid foundation for the project in terms of data that had been previously generated. And again, it comes back to making sure that the student undertaking the project has training in skills that actually help them to go forward. And one of the key skill set that is really important, that is transferable between many different jobs, is really interrogation of large data and mathematical skills in that context. How to number crunch and how to break that down to use the right statistical analysis to analyze that and come up with significant observations. So the solution to actually doing this is to actually have a multidisciplinary research team with defined skills that can help run such a research project. So we actually tried to make sure that we had people that can deliver all of these different elements to try and run such a in silico project. We go to the next slide, please. Okay, so the research team was Aysha Divan. So Aysha is a academic staff member with expertise in cancer biology and signaling. I myself I'm a cell biologist, but with expertise in vascular physiology, signaling, and tumor angiogenesis. And we recruited a PhD student to the team who we actually cast around in the faculty for somebody who could help us. And Isabel was suggested as who could work, and she was recruited as a paid demonstrator who could contribute up to 3 hours per week to support the research project. And she's undertaking a bioinformatics PhD, so she has expertise in database analysis, stats, and various in silico approaches. So the student that undertook the project, Sylvia Ose Owusu. So unfortunately, Sylvia can't be here today. She has other things that she's taking care of. So she was the bioscience master's research student undertaking the project. So what was essential to the project also was to schedule in a weekly meeting, which took pretty much 1 hour. Sometimes it ran for well over an hour, where we went over what progress that was done and discussed some of the experimental or technical issues and problems in carrying out the project. And that's really important to have that a regular meeting at a defined time every week to analyze what has been done and what will be done going forward. So the research student, in this case, Sylvia, had a prior biological sciences related degree, and she had very limited experience of in silico work of bioinformatics. So I should point out again that she thought she was going to do a wet lab project expressing recombinant proteins and doing protein-protein interactions, and it turned out that this was what we could offer her in as a substitution. But she was willing to learn, and that was really an advantage, is that she was willing to tackle new things and actually have a really good go at it. And so, but she had to be reassured that the supervisory team that was in place could help her when she got stuck, especially in some of the complicated in silico problems, the statistics, the training in R, etc. And the end goal was really, what we managed to convinced Sylvia, and we ourselves felt if she could do this, she would actually graduate with a very good degree, and but she would also have a tremendous skill set of learning how to number crunch quite complex data sets, and how to actually break them down and make sense of that. Things that even myself and Aysha didn't know how to do. So that was something that was really, that we spent a lot of time reassuring her that she could do this, and that's quite important. So the project was actually to look at the link between a growth factor called VEGF-A, or vascular endothelial growth factor A, and a transcriptional factor ATF2 that was involved in gene expression, and whether this had any correlation with cancer progression or survival. And the background for this was that for the past 10 years, work from my laboratory had shown in a number of publications that there was evidence for a signaling pathway regulated by a VEGF stimulus that impacted on ATF2 in the nucleus, which controlled expression of a subset of genes in vascular endothelial cells. And this also was actually within the wider context that there are quite a few different studies published and in databases suggesting that VEGF-A could upregulate and downregulate sets of genes in various cell types, especially in endothelial cells, which is one of its main targets. And there was substantial database information in both databases such as GeoProfiles in NCBI, as well as in cancer databases on VEGF-A stimulated or VEGF-linked gene expression. So we were reasonably convinced that there were that there was data sets that Sylvia could analyze and try and see if there was anything going on in terms of the link between VEGF-A, ATF2, and gene expression in a cancer context. Okay, so the background, just a broad-based context of VEGF-A. What we know from work done over the past 50 years is that many solid tumors that actually develop and progress down a pathological route, the pathology is linked to the increased expression of these so-called pro-angiogenic growth factors, such as VEGF-A. And the secretion, the transcription and secretion of these growth factors, such as VEGF-A, acts in a paracrine manner to stimulate stimulate endothelial cells to make a network of blood vessels around the tumor. That provides the tumor with nutrients from the blood to grow, but it also provides an avenue for the tumor cells to escape down those blood vessels and set up secondary growths of metastases elsewhere in the body. But although the role of VEGF-A acting on endothelial cells is really highly studied and very well characterized, the role of VEGF-A acting back on tumor cells in an autocrine fashion is much more murky, and there's little understanding of whether that can modulate disease progression or gene expression or anything itself. So those are two things to be aware of. So what we know is that the engagement of VEGF in this cartoon, VEGF is depicted in these red triangles. It's usually a homodimer, and engagement of the ligand with the receptor, such as VEGFR2 or receptor tyrosine kinase, it can actually impact on many cellular outcomes within the cell. So cell migration, cell proliferation, cell survival, a whole bunch of things are modulated, including gene expression and metabolism. So VEGF-A is one of these multifunctional cytokines that can modulate cell function really on a global level. So in one of the first studies in this, so in 2014 that was published, we showed for the first time that there was a circuit of signaling from the VEGF receptor was via the MAP kinase pathway that impacted on a transcription factor ATF2 that is usually found in the nucleus. And the hyperphosphorylation of this transcription factor resulted in increased transcription of VCAM1. So this is a membrane receptor that actually is then expressed on the cell surface. So that was one of our first published bits of work on showing the evidence for this pathway. So in another couple of studies, we showed that a proto-oncogene called TPL2 also appears to be upregulated by the similar pathway. And this actually impacts on VGFA stimulated tubulogenesis, cell adhesion, and arterial sprouting. In the materials available for the project, based on all this work, we identified a subset of genes that we proposed could be implicated in this pathway. The negative controls to be identified by the student and the supervisory team would then go alongside with these genes that could be interrogated. So the student would be trained in statistics, the use of cancer databases, and profiling gene expression, and building some sort of networks with the data that was obtained. So one of the things that we actually were aware of was to actually try and ensure that all the software or databases were free or easily accessible as academic end users. And one such package that was important was R, which could be downloaded as something called RStudio. And this is a statistical package that can be used to actually number crunch some of these really complex data sets. So there was a whole bunch of references that we could provide, including PhD theses related to this work, as well as providing some sort of, as academics providing some sort of support for the access to the Cancer Genome Atlas and other academically linked databases. Okay. So milestone 1 was training of the student in the use of R and how to use R. And various online resources were available, and the advice from Isabel was really essential for facilitating this, how to get started. Milestone 2 was to make sure the student could register for using these databases and extract the data so that she could then number crunch the data by interrogating that for the link between some of these genes and patient survival or cancer recurrence in these databases. So the support from the supervisory team was essential to see if that she could actually retrieve this data. And milestone 3 was to then do the key analyses, was to, first of all, was to, we then identified colorectal cancer as possibly the most well-known tumor angiogenic state, and then to actually interrogate singly or 2 genes together to see how that impacted on overall survival, or what we call recurrence-free survival, so the recurrence of the cancer. And there are various considerations of age, gender, recurrence, tumor location, tumor grading, etc. So in the first data set was VGFA. So that was a control, a positive control of some sorts. And what Aysha found was that expression of VGFA impacted on overall survival in this colorectal cancer patient data set, but was not but recurrence-free survival was not significantly impacted. So the red line you can see is the reduced overall survival of those cancer patients over time, whereas the blue line is the VGFA low, the red line is the VGFA high. So if you can see the p-value there in the Kaplan-Meier survival curves, that's actually 0.0174. So that's well under the 0.05 limit. So in the next data set, this data set was actually the most astounding and surprising data set, was that, so we put in ATF2 as another thing as part of that axis. And what the surprising thing was, there's a very high link between ATF2, there's a very strong link between ATF2 expression and cancer incidence. So increased levels of ATF2 expression was really detrimental to overall survival. I mean, you can see that p-value, it's really, really low. Whereas it had no impact on recurrence of the cancer. So one of the genes that we interrogated was TPL2, and the single interrogation shows that the overall survival was not significant. In terms of recurrence, that was sort of, the p-value was sort of verging towards significance in terms of whether TPL2 is high or low, but for most intents and purposes, that was borderline. So, so we at the moment we classed it as non-significant. So then we also interrogated. I won't go through all the data. We interrogated 2 genes together, and what you can see is by interrogating 2 genes together, especially things that are implicated in these pathways, that the p-values for overall survival becomes tremendously significant. Whereas for recurrence-free survival, they really don't appear to have any significance. So what we can say is that this has actually thrown up some very interesting ideas about how to analyze genes singly and together in a signaling pathway linked to cancer. And this has actually thrown up a lot of very interesting ideas in terms of a signaling pathway that was originally found in endothelial cells, but that could be operating both in endothelial cells and in cancer cells in terms of cancer progression in these patients. So Aysha, I think if you want to step in and say a few things, that would be good.
Okay, so if I go on to the lessons learned slide, so the student outputs, so the student generated actually some really valuable data sets, some very unexpected data sets, and some very valuable information. In terms of her assessment for the Masters itself, she was expected to submit a draft manuscript in the form of a scientific report and present in the form of a research poster and attend a viva. So she's in the process of going through this. And we do feel that actually the work is of such a quality that it could lead to a publication with some additional work that could be completed subsequently. But in terms of the lessons learned, so Vaz and I both said that we didn't really have a lot of experience right at the start in terms of conducting or supervising computational projects, and neither did the students. Overall, both the supervisors and the students enjoyed the project and really extended their existing skill set. One thing we would say is that the supervisory team needs to be selected quite carefully so that there's the right type of skills, appropriate mix of skills to reassure a student who particularly may have to conduct large pieces of the project online, be computational with skill set that they don't really have and are trying to build up through the project. And we did find from the students' perspective, so the project itself was about 4.5 months. We did full time she was working on this project from her home. We did find actually that the first couple of months at least was all about learning how to use R, registering for the database, working with sample data set, and just trying to understand what all mean and how to carry out meaningful interrogation and analysis and interpretation of the data. So if this project was adapted, for example, for a shorter project, a shorter time frame, then I think there would have to be some consideration of the lead time for learning of new skills. And that could be, for example, through intensive training upfront rather than on the project. That might be one way prior to the project starting. We've also considered the possibility of how would this work. So this was a individual students working on a project. We've also considered the possibility of how would this work if this was offered as a group project to a group of undergraduate students. So multiple students working on the same problem or a similar problem, but with different data sets using the same analytical techniques. And we think it could work rather well, actually. It would allow students to support each other in skills development, but also share some of the data sets and perhaps have fun on a particular area in terms of building models and hypotheses going forward. So overall, I think it started off, we had to transition very quickly in a very short period of time. But I think overall, with quite a lot of, say, planning and preparation and encouragement, the student did become very independent as the project progressed, generated lots of very useful data, of which we've only shown you a subset, and could lead to something publishable moving forward. So I think we were very pleased, and I think the student was also satisfied that she had received a positive experience of a project. Not quite what she was expecting, because she certainly was expecting a lab-based project, a wet lab-based project, but the computational project gave her some valuable training to take forward as she moves into the next part of her career. So I think we'll stop there and take questions.
Thank you very much, Aysha and Vaz. That was really interesting, and we have generated a lot of questions from there. Thank you very much for the attendees for staying with us. Obviously, we had a few technical issues at the beginning of the process, but I think we managed to resolve those on the fly. And those of us who've been working remotely for some considerable time will realise just how frequently these sorts of things crop up. So again, thank you for bearing with us. We do have a number of questions that I'm happy to put to Aysha and Vaz. As it says on the screen, if you've got a question, type it into the questions box, and then we'll go through them in a reasonable way as we can. I'll try and group these into sets, if that's okay, based on topic areas. So again, thank you for everyone who's already submitted something. But if you have got any other questions, please keep those coming in. Many of the questions are perhaps not unexpectedly around more generalizations rather than the specific topic areas, but really around trying to make sure that individuals can set up projects of this sort, bioinformatics projects and other things, and how students are able to benefit from them. So the first question, which might be for Vaz possibly, but is what advice would you give to students as to how to make the most of in silico projects?
I think how to make the most of it is to actually, I think, undertake that really hard training up front. It's the grappling with the databases and the R. I mean, I think that to look at it and think of the bigger picture going forward, if you're going into a PhD, or if you're going into, well, even I think if you're going into the city as a stockbroker or any sort of job, the ability to grapple with complex data sets and large amounts of data and numbers, and if you can break that down using a package such as R, which is a freeware and easily available, it actually places you in a very good position to actually get really good employment in the long term. And I think that is really so. But what we were very pleased with Sylvia is that it was tough for her at the beginning, and but she really stuck at it. She really persevered, and she didn't quit. And that was something that both me and Aysha were very pleased with her and very proud that she stuck through it through some very tricky, tough times, working alone in a room on her own on campus, and really persevered. So it would be quite tough, and they've just got to stick at learning it. And I think it's really important to have somebody in the team. We had Isabel, who could actually provide that expertise in terms of ironing out I mean, she downloaded various things, pointed her to courses in Cambridge, freeware, seminars, and a whole bunch of things that really added to Sylvia's experience and made her feel not alone in terms of getting help and that thing. And that was something that me and Aysha couldn't do because that was just not our area. So it's really important that you recruit somebody who can do this practical management. And so we paid from our budget, we made the case to the school, and we paid Isabel as a demonstrator for the amount of time that, so she kept a log of the hours that she spent per week on Sylvia's thing, and she was paid, which was only fair, I think. And so she was paid per week for time spent, and that turned out to be a very good move. So I think I would say try and recruit somebody and make it, I mean, I think if you recruit another lecturer or a PI who can do this, that's great, but you want to make sure that they will be there and they can put the time in. I think by luck than anything else, having a PhD student committing that time was actually, I think, far more useful than anything else. Aysha?
Yeah, that's really helpful, Baz. Thank you. If I could bring in another question, because it's based on the back of your answer, really, which is how much hands-on training in the software or statistical packages did the student need?
Zero, I think. I mean, I certainly, I mean, I know how to use GraphPad, and that's about it. So, so I think, you know, we had a theoretical view what we wanted to do, and we just sort of very grandly asked the student to implement it, which is sort of typical for many lecturers, I think. So, but the amazing thing is that Sylvia rose to the challenge amazingly, and she did a fantastic job. So I can't speak more, you know, more highly of Sylvia. So I think, but I think like everything else, I think at this level, if you set somebody a stiff challenge, most people rise to it, and actually, but they have to be convinced that it's going to also help them going forward. But I think she did rely on people to. She actually emailed another faculty member in medicine, Mark Islesworth, and just sent a few questions here and there to various people and things like that. So you need to make sure that there are enough people around to actually give sorts of advice and help, and that is available. And I think in that sense, the academic community is really good to deliver that.
Aysha, is there anything from your perspective?
Yes, so I think that's right. So Sylvia's starting point in terms of statistical, mathematical skills and bioinformatics skills was fairly fundamental, I would say. So she was certainly very well trained in wet lab skills, perhaps less so in computational skills. So I think that first part of the project, when students are expected to learn and then become independent, I think that requires quite structured support. And if that is right and the student approaches that and engages with that well, then I think the other parts become easier. And I think that's partly the role of the supervisor to make sure that that happens.
That's really helpful, thank you. The next question is, were online lectures available to the students to provide bioinformatics support?
So the students had actually completed, so these particular students, the cohort that Sylvia derives from, do have a 10-credit bioinformatics module that they take as part of their TORS MSc. So she did have some skills, and that particular module is delivered in semester 1. It includes a combination of lectures, practical sessions, so computer-based practical sessions, and then obviously associated assessments with that. And she had completed that successfully. And the module manager and the staff associated with that would have been able to provide her with some additional, more advanced level support if we'd approached them. I guess our approach was that we allocated a PhD supervisor to support her in that part.
That's really helpful. We've got a couple of questions around data and availability of the data that I think would be useful to go through there. So the first question is how much internal data had to be provided to the student?
So, so I think in terms of, I mean, what we provided to the student was really absolutely no internal data. It was only stuff that's been published. We just provided her with lists of genes that we thought could be involved. And that was from work that had already been published. There was some unpublished work, but really that turned out to be not really that useful. But anyway, you know, most of the stuff been published, and really there was other stuff that had been published and stuff in also in geoprofiles and in the TCGA, that loads of data that was unpublished but freely available for mining. And so that was really the goal for her to access that. But we didn't give her any hard data apart from the list of genes that we thought may or may not be involved.
Thank you. And on a similar topic, the question is, projects of this kind are very dependent on the quality of the data in the databases. Do students and supervisors have enough information about the experimental design that generated the data?
Yeah, I mean, that's a really good point. So she actually identified a single data. So she worked with one data set that was a colorectal cancer data set. So originally we thought she could interrogate multiple cancer data sets, but that turned out to be basically there's not enough time. So she focused in the ends on colorectal cancer because that was potentially the most tumor angiogenic state, and there's actually anti-VEGF-A antibodies used for cancer treatment in that particular situation. So it was clinically relevant as well, and that's why colorectal cancer was the obvious thing. But she actually ended up looking at a single data set. There wasn't enough time for her to look at anything else, but she waded through quite a few different things until she found one data set that she could work with that had enough information. And that data set we presented was based on about 60 odd patients followed over a 5-year period.
Thank you. That's really helpful. A couple of questions around the PhD students that was providing the instruction. How easy was it to provide prompt help to the student if the PhD instructor was busy?
So we had so Isabel was recruited. We defined upfront what her time allocation was. So we said as a PhD student, she could spare a maximum of 3 hours supporting Sylvia on her project per week. And I think at the start of the project, that probably was 3 hours that she was spending time with Isabel, and Sylvia were spending time together, I think. I mean, some of that time will be spent kind of doing other things, but 3 hours, and then that decreased as Sylvia became more independent. I think they managed that between themselves. And I think the expectation was that there would be email communication. So Sylvia could email Isabel, the PhD student. She could email us as well with questions. But there would be defined time when the two of them, without Vaz and I getting together, where they could share screens.
I think we need to stress that Isabel is not a PhD student with either of us. Isabel works for informatician David Westhead, and we worked out with him what was sensible and allowable for Isabel to commit per week. And so so we had to work that out in the background before Isabel was committed. So that was part of the management we had to do. So 3 hours per week. I think in the first few weeks, Isabel might have done like 5 hours per week, and then it dropped down significantly as Sylvia became more and more sort of au fait or experienced with all these manipulations.
On a similar kind of vein, the next question is, will Isabel be included as an author on any potential manuscripts that
definitely, and a supervisor.
I assumed that was going to be the case, but just thought I should check.
Well, if nothing else, we can't make sense of the numbers. We need her expertise.
Fair enough. Another question is, how much help in the writing of the report was provided by the supervisory team?
So Sylvia is still writing up her project. She's in the process of doing that. So the idea with master students in biological sciences is because we have weekly meetings, we look at the data quite closely and discuss it with the students, and they write up their methods and so forth as they go along. She will submit a draft to us for comment in the next few weeks, which we will return to her prior to submission. So it's a fairly independent process in terms of writing it up. But we have had very regular meetings that feeds into that write-up process, and then a draft that we comment on before final assessment.
Sounds good. Sounds good. A question about scalability. So how does an approach of this sort be scalable? It seems relatively resource intensive for staff and the supervisory team. Do you have any thoughts about how you could potentially scale it to, I guess, to undergraduates, but then to a wider range of students?
So I think me and Aysha have discussed this, and this is a really interesting question. So we suggested that going forward, because we still don't have, until December, we're not allowed students in the lab, and we don't know in January whether we can get the students back in the lab. So going forward, it may be possible, I think, whether it's undergraduate or masters, to actually have potentially a group of students sharing the same platform, but with slightly different questions or endpoints that they're aiming for, different subsets of genes or different things. So they might all start off as a group of 5 or 6 students or even more, learning common things like R, statistics, whole bunch of things, and feeding off each other's. So they could work, I think, as a group, but they might have much more specific questions unique to each student. So I think there is a way of designing so that it's actually much larger, but also specific to each person, rather than providing the same research question to all the students in the group. So I think there is a way of doing it, and we are thinking about how to do that right now.
That's really helpful.
And also, I guess it's also worth adding that we transitioned to this at very short notice. Whereas if there was more planning time, you could do exactly what Vaz suggested. And I think we suggested in our slides right at the end as well, which is think about ways that you can provide training in certain skills upfront to larger groups of students, so more teaching of skills, and then progressing them to the project element. I think that would certainly help.
Thank you. This is probably a question for Aysha, because I know that she and I have discussed this before. But how might we best recognise recognise or mark the group work elements in the final dissertation? This is kind of taking things a bit more widely, and what weighting might be appropriate?
Yes. So I think when we were thinking about group projects, so the idea would be that everybody does have a distinct set of work that they need to do. So there is a defined element that they have to work on, even though they might be sharing ideas and sharing kind of conversations about what it all means. There might be an element of where they pull the data together and say collectively, what does this tell us in terms of the bigger picture? But I think ultimately we would be looking at the write-up being independent, so that we could clearly identify exactly what the intellectual contribution of the particular students would be, and the output would be looking at, certainly from our perspective, I think we would be saying the output has to be individual.
Thank you. A longer question, but please bear with me. So what would your opinions be on the development of a postgraduate module which would focus on the design and delivery of projects like this and give the students recognition in connection with credit accumulation for structured PhD programs?
I think it's an interesting question, actually. We hadn't thought about it, but I think you could say, are there training elements associated with this, and then some original work that goes with it? What do you think, Vaz?
Yeah, that's an interesting idea.
Yes,
So
hadn't thought about it.
yeah, I mean, I think you'd have to try it and see. So it might be quite useful to do that as a way of students trying to identify their question and actually their project as they go into it. And that might be perhaps linked to some sort of bioinformatics training as part of the module. And then they develop that question or the in silico project, and then they are assigned a supervisory team. So that could be an interesting way of doing it, and perhaps more structured, and it might sort of throw up a lot more interest. But I think you'd have to have a whole bunch of different questions or things that different people could pick up and run with and sort of work with and sort of just shape and develop. So it could be as part of, you know, developing a project proposal and things like that as part of the module.
I think that's right. I think, as Aysha said, it's creating that independence for each individual student, isn't it? So they may be doing a similar project or topic, but it's creating that individual dissertation that they can write. Sorry, Aysha, I think you were going to say something.
Yes, I was just going to say, actually, one of the challenges I think with computational work, real challenge, is how many people are there with the right skill set to be able to deliver or support this type of project. I think that's really one of the things that we struggled with. And I think when it comes to the question of scalability, I think we've got to look at that quite carefully in terms of how many people within the organisation and perhaps out with the organisation could be brought together to support projects of this type. And I think we'll find, because really what we're trying to say is these are people like myself and Vaz trained in wet lab science who are now having to learn new skills. But the project supervisors need supporting and the student needs supporting, and how many people are there that can provide a pool? So can we provide a sufficient size pool for these types of projects so that they can actually happen in a meaningful way for the students?
That's
and I think
helpful.
that's a real challenge.
Indeed. Another question is on a more technical level, which is, would it be useful for students to prepare by taking courses in R and statistics? It says here on MOOC platforms, but those of us who've been involved with the Biochemical Society know that we run R for biochemists as well as an online course. Do you think those sorts of things would be helpful, or do you think there's enough kind of on-the-job training as students doing the projects?
Definitely. I think R is going to be, I think different universities are starting to implement R in their degree programs at different levels. So, I mean, we haven't used R routinely in our, whether it's on a postgraduate, undergraduate programs, but we are starting, I mean, me and Aysha are starting to discuss this very intensively and take this forward, because I think that we need to probably bring R even in the late stages of an undergraduate teaching into play, and to try and get the students much more statistically aware. And I think because of the issues with using Excel, SPSS, and GraphPad, various issues, I think R is, if you ask the mathematicians and the people in much more of the physical sciences, they all say that R is the way to go in terms of number crunching data. So I think that R is really and right now the best choice for manipulation of large scale data sets. And learning how to do it, I think, is a really important skill for people in the life sciences.
Thank you. We've got a fabulous question now, which is one that I can fully appreciate, which is when I look at the various databases, such as the Cancer Genome Atlas, they are just overwhelming, and I wouldn't know where to start in terms of guiding a student. Would you consider publishing an idiot's guide to how to navigate the database, and what questions you can ask and what approaches you could take?
Yeah, I mean, I think that, yeah, that was for us also very, very difficult. So we sort of devolved that down to Isabel and Sylvia, really. I mean, we sort of set the sort of context, and really it was up to Sylvia to work her way through different data sets and to decide, to make some judgment calls which data set was the best. We had some advice from various cancer biologists and things like that, but there wasn't much to go on. So the only way is to actually look at the different data sets. I mean, it may be that once you define your particular cancer or disease phenotype, then to get some advice from somebody working in that field, what is a good data set to work with. I mean, it could be, I mean, Sylvia just had to wade her way through this different things until she identified a single data set that she felt was good.
But you could imagine if there was prior training built into undergraduate degree programs, even at the, say, higher end, you could provide example workflows for students to work through, and that provides some training, and then they're able to transition to more independent work in a project. But I think kind of a dummy's guide is a good idea. I think it's a useful suggestion, actually. I think there'll be lots of people who would find that type of documentation useful.
Yeah, I mean that was, I think when we started this, that was one of my hopes, that at the end of it there was a idiot's guide for me to do it. I don't think we have it yet. Again, I look at the databases, and like any person out there, I started hyperventilating and my blood pressure starts rising. So I think Sylvia actually and Isabel, between them, actually did a fantastic job of slicing it up and wading their way through it. I think without Isabel on the team, we certainly couldn't have done this.
That's really helpful. Thank you. I'm aware that time has almost beaten us. I would just like to extend my thanks to both Vaz and Aysha for really interesting discussion, and obviously sharing the information that they've obtained during the supervising of this project and the work that they've done at Leeds. Also, I'd like to thank all of the attendees for their attention. You know, there are lots of issues in which you could get involved with on a Friday afternoon, and it's greatly appreciated that you took the time to be with us today. These are clearly new and challenging times, and it's more important than ever to stay connected. All of us who work in universities or laboratory settings realize just what an extraordinary time it is. But it's also an exciting time to join the Biochemical Society and to take part in activities like this, and joining communities of researchers and specialists, and to be able to stay connected. So we welcome suggestions for future topics for webinars for this series. Our next one is on September the 17th, which will be co-hosted by the British Society on Research on Aging, and the title of it is going to be Genes Regulating Aging and the Quest for Immortality, and you can register for that on the Biochemical Society website in the same way as you did for this one. So thank you again to our speakers, thank you to the audience. I wish you every success in your work or in your studies, and thank you for joining us, and I look forward to being able to welcome you again to one of these events. So thank you all very much, and please have a good rest of your days. Thank you.
Thank you.